resilience in protein interactomes across the tree of life.
Sorry. Hello, everyone. Today I will talk about evolution of resilience in protein interactomes, and this is joint work with Mark Feldman from Stanford Biology and Yuri Liskowitz from Computer Science Department. And the way I will start is really by thinking outside of the biological context. In particular, if you think of airplanes or systems, for example, complex systems such as an airplane, we can see that there's a number of parts and components that need to work together in a predefined way for the airplane to function properly. And engineering advancements in engineering in the, since 19th century have really enabled the planes to evolve at such a rate that they can do much more now than they were able to do at the beginning of over 100 years ago. So really analogously, there in human cell, there is this additional other kind of wiring diagram where we have protein interaction networks, the way proteins interact with each other, that really defines the backbone of activity in the cell in a similar way as we have those physical parts and components in the planes. And what is really then important to understand, and an important question to ask is, well, how do those components, molecular components in a cell, how do they interact and how do their interactions evolve over time in a similar way as we might be interested in understanding how do those interactions between physical components of an airplane have changed. And really then the main question that arises is, well, how do protein interaction networks evolve? How do they rewire over time? And that's really an important question to which we don't have yet a very good answer. And it's not really clear whether or not and how really natural selection shapes the evolution of protein interaction networks, and that still remains unclear. So in today's talk, I would like to provide some insights into and some initial answers to these questions by focusing on two specific questions. In particular, we will try to answer and provide insights into how protein interaction networks change with evolution, and how those network changes really can affect phenotypes, and what is their impact on organisms' abilities to survive in certain kinds of natural habitats. And there are a number of reasons why modeling network evolution is challenging and hard from the computational point of view. And I will mention briefly three kinds of challenges. So first is certainly that there is massive time span and rare data samples. What I mean by that is that species are separated by millions of years of evolution, and what we have really is information on interactions between proteins of present-day species. We have little to or no information on interactomes or interactions in species that are extinct. Furthermore, the data that we have is messy, incomplete in many different ways, and there is lack of high coverage protein interaction data for a number of species, including popular model organisms. And because of that, there are many possible confounders that one would need to take into account when trying to approach the problem of really understanding the evolution of networks from the computational point of view, since there are possible confounders that arise from the biological factors, such as the kind of platform or assay that was used to generate the data, as well as those biases that arise from non-biological factors, such as investigative biases that we see in the data. So our approach to tackling this challenging question consists of two main steps. So in the first step, we constructed and built a data set by integrating and combining data on species-specific protein interaction networks, their evolutionary information of species, and ecological data on natural habitats in which species live. And secondly, we will use that data to really study the evolution of networks, in particular asking ourselves how do those networks change with evolution and what kind of impact that they have on species survival. And really the first key element here is to have this high-quality data set that we can construct based on the data that is out there currently available. So for that, we built a data set on species-specific protein interaction network, where we primarily relied in many ways on the wonderful resource provided by String and other consortia, and integrated that with information on evolutionary history of species and complexity of habitats in which species live. The way you can think of this data is as a large tree of networks. So what I mean by that is that in the center of this slide, you see a phylogenetic tree, which represents the evolutionary information and relationships between species. And at the tips or leaves of that tree are present-day species for which we have some amount of protein interaction data available. So it's a tree of networks where those networks, which are partial protein-protein interaction networks, which we will call interactomes, are positioned. And we will represent every species as its own by its protein interaction network. And for example, here we see two specific networks, one for eukaryotic species and one for bacterial species. So given this data, our task is, well, we want to study how those interactome networks change with evolution. In particular, the main question that we'll be trying to understand better is how do those networks respond to various kinds of protein network failures that we can see in the interactomes. And it's really the reason why we focused on this question of understanding the robustness and resiliency of networks to failures is fundamental, because the breakdown of proteins interactions can have many detrimental effects on organisms, and is associated with a number of unwanted effects and diseases. So we really want to understand this better. And to understand and characterize resilience of protein interactomes and resilience to network failures, we define a new measure, which we call interactome resilience measure, based on Shannon's diversity theory, which I will explain in the next slide. And this measure has three main components. In particular, for a given protein interaction network, we will first simulate a protein network failure at a particular rate, and then we will measure what is response of the network to that failure rate. And then we will do that. We will repeat this process through all possible failure rates and integrate that in order to get information on the resilience. So what I mean by that is the following. So here on the left, you see a hypothetical interactome where nodes indicate proteins, edges represent physical protein interactions. Upon a specific network failure, which is represented here by randomly removing a set of proteins and all of their interactions, the network will become dissociated and will fragment into a number of isolated components, shown here by these gray blocks or regions. Now, what is really important is to understand into how many isolated components that has the network broken into, and what is the size of those components. A natural way to characterize that is through entropy on the size of the connected components, which is shown here on the right part of the slide. So to give an example, here you can see two synthetic toy interactomes on the left and on the right that have the same number of nodes and the same number of edges, meaning interactions. And in both of the interactomes, we have also removed the same number of nodes. So we simulate failure at the same rate. But the response of the two networks to failure is vastly different. We can see on the left, the network has really broken into a number of small isolated components that are all of approximately the same size. And the way we can see or characterize that quantitatively is by really getting high entropy value on the cluster component sizes. On the right, in contrast, even though we remove the same number of proteins in their interactions, we still have this relatively large connected component, and because of that, there is low entropy on the distribution of disconnected components. So this is what we got from the previous slides is really quantification of response of the network to a particular failure rate. But really, to get the resilience, we need to resilience of the interactome, or we need to integrate the response of the network across all possible failure rates. And that gives us insight into how resilient the network is by really seeing whether it dissociates fairly quickly to a number of disconnected components, or it stays relatively connected even for high failure rates, which would indicate a highly resilient interactome. So this is an approach that we then employ for every interactome and every species, and this gives us this outline bar around our phylogenetic tree, which is shown here as, in the form of gray bars. The question then is really, how does that relate to evolutionary information? And here is our first finding. So first, we find that protein interactomes are becoming more resilient with their evolution, meaning that more genetic change a species has undergone, more resilient is its interactome, and further indicating that those interactors becoming more resilient to failures over time. But I guess many people in the audience now have already asked, well, what about all kinds of potential confounders that I was mentioning at the beginning of the talk? And really to study that and answer this finding is due to solely data biases, we devised a causal model that allows us to really understand whether that relationship between evolution and the interactome resilience that I showed on the previous slide is due to actual properties of the data and the potential underlying biology, or if it can be solely explained by various biological and non-biological factors. For example, simply the amount of research that has been done on a particular species, or the average degree of proteins in the interactome networks. So we find that our key findings are not due to data biases and cannot be explained by a number of possible confounders, and furthermore, they are consistent across taxonomic groups and robust to network data quality, network science, and the kind of biological assays that are used to generate the data, which indicate that key findings will still hold when more protein interaction data become available. So now we know that those resiliency of networks increases over time, but does that have any useful impact or effect on organisms? So it turns out that resiliency is very beneficial in terms of that organisms with more resilient interactomes can survive in more complex, more diverse, and more competitive environments. So in this plot, I'm showing the interactome resilience on the y-axis, so higher value indicates the interactome of the species is more resilient, and on the x-axis are various aspects of natural habitats in which species live. And furthermore, we can also characterize, well, how competitive are the environments in which species live, and how hard it is for species to fight with others for the limited amount of resources. And again, we see that, that species with more resilient interactomes tend to thrive in those more complex, more variable habitats and are able to survive in those. Well, this is really an observation at the global level because we have this notion of resiliency of interactome at the entire network level. Now, the next question is, well, what are those local network changes that would give rise to a globally resilient interactome? So in order to understand that, we're thinking about local interaction neighborhoods and trying to understand, well, what happens when we see a speciation event and a species evolves into two new species. In the protein interaction or the network sense, the current model that is most commonly used to study that is known as a duplication and divergence model, where the idea is that we have in this hypothetical tree where there is a speciation event here on the left, a particular protein A in ancestral species will copy itself, so we'll have A prime in the newly formed species 1 and A double prime in species 2, and now those proteins can rewire their local interactions in each of these species over time. By rewiring, I mean that the species might add a new interaction, it might remove a particular interaction, so different kinds of things can happen. Really then the question is, well, how do those interactomes rewire over time? And to study that, what we did is for this large data set, we constructed local protein neighborhoods known as ego networks around every protein in every species, and then we studied their connectivity based on orthologous relationships. So it turns out that resiliency arises through gradual change of network topology. In particular, what I'm highlighting here is if we start with this with the A prime protein, its local network neighborhood, over time that local interaction neighborhood will become more connected and more redundant. So here what I'm showing in gray is how many isolated components do we have in that local neighborhood if we remove A. Here we have a bunch of those isolated components, but here in a different species that has seen more evolutions, those components are fewer and they are much larger. And we can quantify that in a very principled manner and compare with various null models, showing that those local interaction components are becoming more connected and redundant over time, which gives opportunity for the networks to become more resilient at the global level. In addition to that, there are a number of mechanisms of resiliency that we can identify. What I would like to point out here is really studying then not only pairwise interactions, but higher order network structure or the rewiring of network motifs. And what is interesting here is that we see that square network motifs are becoming more common in protein interaction network. And there's a range of biological evidence that would support this finding. And the reason or the one mechanism to which those square network motifs can emerge is because proteins with similar interfaces can share many interactors, many neighbors in the networks, but they do not necessarily directly interact. And this naturally creates those square types of motifs. And the analogy here is thinking of having a key lock and a key, so they need to fit together and have the similar interface for the proteins to interact. So to summarize, the key new insights here are that resilient interactome is a measure that indicates that proteins are able to interact with each other in the face of network failures. Those failures might be neutral in a current environment, but those might not remain neutral indefinitely. So when the environment changes, all of a sudden the power of resiliency can really offer the species opportunity to thrive in a changed environment. So the way to think of it is a resilient interactome is a reservoir that drives future evolution and gives opportunities for evolutionary innovation, which has a bunch of consequences and implications for design of robust systems and as well as ecology. And my last slide is just I would like to shamelessly make a remark that I'm hiring postdocs starting later this year at Harvard, focusing on problems related to network biology and machine learning for graphs. Thank you.
Thank you very much for that exciting talk. We have time for 1 or 2 questions. Yes.
Hi, thank you for your talk. Very interesting work. I see that you compare unicellular organisms and multicellular organisms. I wonder if there is any kind of effect in the network topology considering these different organizations, and if you consider context-specific networks like tissue or organ-specific. Thank you.
Yes, these are great questions. So first, regarding multicellular, cellular, and unicellular organisms, there's a number of networks differences in their network structure, which we studied quite extensively in the paper, and I was not able to cover here. And really, to understand this better, what we did was to break our analysis by taxonomic groups, so that we were then able to see whether our findings at the global level are also true within a specific taxonomic group or within a specific group of organisms. And the answer to that is that yes, the key findings are still can still be seen there if we only focus on the multicellular or only on the unicellular organisms. Regarding context-specific networks, we have recently been working with tissue-specific protein interaction networks, and we've been applying methodology there, but we're not studying tissue-specific networks within this work. But it's a great opportunity for future direction.
Thank you. I have a technical question and a more broad question. Technical, how exactly did you define the evolutionary time? Could you be specific how you did this? And the other question, since you have this tree of networks, and even showed it, I guess you made the tree on the sequence similarity, or is it basically if you compare your species similarity based on sequence similarity and network similarity, is that a very good match, or can it be quite different?
Yes. So these are great questions. So to first answer the question that you have, or let me start with the first. The evolutionary time in the plots that I have shown here is defined as the total branch length from the root of the phylogenetic tree to the leaf. According to the paper published in Nature Microbiology 2016, the new view of the tree of life. So the phylogenetics tree there are constructed based on DNA sequences, but also in addition, they are also providing the tree constructed based on protein sequence. We also studied a number of different alternatives, or used different alternatives, phylogenetic trees that are out there to see whether our findings would be still true, depending on different ways to defining the evolutionary time.
In terms of time, I think we might ask you to find Marinka in the break, so we stay on track.